All right, let's just dive right in. Welcome to today's explainer. We usually picture a cell as this kind of flat, static blob on a textbook page, but today, we are completely flipping that perspective. We're going to take a tour of the cell as a bustling, microscopic metropolis. It is literally a living factory, and it's operating 24/7 inside of you right this very second.
But before we can even step through those factory doors, we've got to talk about scale. Like, what actually happens when things get smaller than 0.1 millimeters? Well, it turns out 0.1 millimeters is the absolute limit of resolution for the human eye. Just imagine two tiny dots drawn on a piece of paper. As you move them closer and closer together, once they cross that 0.1 millimeter threshold, your eyes just can't distinguish them as separate anymore. They just blur into a single point. So to really see our factory, we have to go way beyond human limits.
Just look at the massive jump in scale we're dealing with. We start up here with a human, shrink all the way down past a chicken egg, past a tiny amoeba, and drop right into the nanometer world. And it's wild to think it wasn't until 1665 that scientist Robert Hooke finally pierced this veil. He used a self-designed microscope with, about 300x magnification, and looking at a simple slice of cork, he spotted these tiny box-like compartments. He officially dubbed them cells.
Fast forward to today, and obviously we know these cells are incredibly sophisticated. Like any major factory, a cell needs intense security to control what comes in and what goes out, right? That security gate is the plasma membrane, and it relies heavily on a really fascinating biological bouncer called osmosis. Basically, osmosis is constantly moving water through a selectively permeable membrane, shifting it from an area with more water to an area with less water. It's doing this to maintain the perfect balance so our little factory doesn't flood or totally dry out.
And we can actually see this microscopic security system at work using something as simple as a kitchen potato. If you drop a piece of potato in plain water, which is a hypotonic solution, the cell membrane lets water rush into the cells and the potato swells right up. But if you take another piece and drop it into a 20% salt solution, so a hypertonic environment, the water rushes out of the cells trying to dilute all that salt. As a result, the potato visibly shrinks and gets super limp. It's all just cells reacting, swelling or shriveling based entirely on osmosis.
Now, while all cells use these membranes, their outer defenses vary wildly depending on how the organism actually lives. For example, animal cells have this flexible fluid mosaic membrane. And that makes sense, right? Animals need to run, jump, and bend, so our cells have to change shape easily. But plants? They can't exactly run away from a storm. So plant cells are reinforced with a rigid outer structure called a cell wall, which is mostly made of a super tough carbohydrate called cellulose. This extra layer is exactly what lets a huge tree stand tall against heavy wind and rain without its cells just bursting from absorbing too much water.
Okay, let's step past those outer walls and right onto the bustling factory floor, the cytoplasm. This is where we find the organelles. And just like a real metropolis, everything here is brilliantly compartmentalized by its specific job. You've got mitochondria acting as the powerhouses, pumping out energy. Lysosomes? Those are your biological waste disposal units, breaking down debris to keep the whole place clean. And vacuoles act as these massive storage warehouses, especially in plants, hoarding water, minerals, and nutrients to keep everything firm.
Let's zoom in a bit closer on those energy generators. Take the mitochondrion. Look at its inner membrane. It's intricately folded into these finger-like projections called crista. Those folds massively increase the surface area for the chemical reactions that generate ATP, which is basically the energy currency for almost everything the cell does. And then you have the chloroplast, which is an organelle you'll exclusively find in plant cells. These guys contain chlorophyll and act like highly efficient organic solar panels, capturing light energy to literally synthesize food out of thin air.
But of course, every single factory needs a central command center. And for the eukaryotic cell, that headquarters is the nucleus. When we peek inside the nucleus, you'll see this tangled thread-like mass called chromatin. That is your DNA. I know it looks kind of messy, but right before the cell is about to divide, that tangled chromatin perfectly organizes itself into these distinct rod-shaped chromosomes. Those chromosomes hold the master blueprints for the entire next generation of cells.
Although, there is one totally mind-blowing exception to this. Not all cells get to keep their headquarters. Mature human red blood cells actually eject their nucleus entirely. They literally sacrifice their command center just to make more physical room for hemoglobin, which lets them transport way more oxygen around your body. But, this sacrifice comes at a pretty high cost. Without a nucleus, they don't have the blueprints to repair themselves or divide. So they have this strict, non-negotiable lifespan of exactly 120 days before they die. So the takeaway here is that while animal and plant cells are highly organized, not all life shares this tidy structure. Just take a look at a typical bacterial cell. This is what we call a prokaryotic cell. It is drastically smaller, and it completely lacks a well-defined nucleus. Its genetic material just sort of loosely floats around in an unenclosed region called the nucleoid. It's a primitive design for sure, but incredibly effective, and it's a really sharp contrast to the highly compartmentalized plant and animal cells. Yet despite all these structural differences, every single one of these cells is bound by a foundational set of biological laws. Back in the 19th century, scientists like Schleiden, Schwann, and Virchow pieced together what we now call the classical cell theory. It all rests on 3 pillars. Number 1, all living organisms are made of 1 or more cells. Number 2, the cell is the fundamental unit of life. And number 3, which is honestly the most profound, all cells arise from pre-existing cells. Life literally only continues because existing factories physically divide to create new ones.
And the coolest part is, we can witness this replication happening in real time. What you're seeing here is an incredible microscopic view of an onion root tip. Since a root has to constantly push and grow through the soil, its cells are rapidly dividing. You can clearly see the different stages of mitosis playing out. One parent cell is meticulously copying its genetic blueprints and then cleanly splitting into 2 genetically identical daughter cells. This exact process of mitosis is how a plant grows, how you grow taller, and how your skin magically heals after a paper cut.
But we also have to talk about what happens when this replication goes rogue. Normal animal cells have this brilliant built-in safety mechanism called contact inhibition. Basically, when dividing cells come into physical contact with their neighbors, they get a signal to just stop dividing. It's a perfect balancing act. Cancer cells, however, lose this vital control. They completely ignore those contact signals and just divide uncontrollably, stacking on top of each other, which is ultimately what forms a tumor. It's literally the factory's machinery stuck on overdrive with absolutely no brakes. Which actually brings us to a modern breakthrough that kind of challenges everything we just talked about. Remember rule number 3 of cell theory? That all cells must come from pre-existing cells? Well, back in 2010, scientist J. Craig Venter and his team pushed the absolute limits of that rule. Using computer programming, they chemically synthesized the entire DNA sequence of a bacterium from scratch in a lab. They then took this man-made synthetic DNA and inserted it into a pre-existing cell that had been totally emptied of its own genetic material. Astoundingly, the cell booted up, it survived, and it began to divide based entirely on those synthetic instructions.
Now, sure, they didn't build the entire physical shell of the cell from scratch, but they did write and install the software of life completely synthetically. And that leaves us with a really profound question to wrap up our journey today. If we can literally type out the DNA sequence of an organism on a computer, synthesize it chemically in a lab, and use it to control a living cell, what exactly defines a living thing? Where's the true boundary between mere chemistry and actual life? Keep that thought in your head the next time you think about the trillions of tiny, living factories working inside you right now. Thanks so much for joining me on this explainer.